Hello everyone, welcome back to our channel Biodiversity and Conservation. I am Nivedita Chenna from Ambika Prasad Research Foundation. Today going to discuss about therapeutic proteins, its classification, function, with future directions. So let's start. So proteins which are engineered in the laboratory for pharmaceutical use are referred to as therapeutic proteins. And proteins which are absent or low in individuals with an illness such as cancer, infectious diseases, hemophilia, anemia, multiple sclerosis, hepatitis B, etc., are artificially synthesized on large scale through genetically modified host cells and delivered. This therapeutic approach in treating diseases using proteins and peptides is termed protein therapeutics. Protein therapy is similar to gene therapy, but unlike gene therapy, protein therapy delivers protein to the body in specific amounts to help repair illness, treat pain, or remain structures. And it was introduced in 1920s. Human insulin is considered to be the first therapeutic protein. So then coming to its functional type and pharmacological activity. In enzymatic or regulatory activity, the pharmacological activity is replacing a protein that is deficient or abnormal. For example, insulin for diabetes mellitus, growth hormone for growth failure, and coagulation factors for hemophilia. And other pharmacological activities augmenting an existing pathway and providing a novel function or activity. In special targeting activity, the pharmacological activity is interfering with a molecule or organism and delivering other compounds or proteins stabilizing unstable proteins. In the vaccines, protecting against a deleterious foreign agent, for example, hepatitis B surface antigen for hepatitis B vaccination, and treating an autoimmune disease and treating cancer. And diagnostic function type, in vivo infectious disease diagnostics, for example, recombinant purified protein derivative for diagnosis of tuberculosis exposure, and hormones like glucagon as diagnostic aid to slow gastrointestinal, and imaging agents, for example, capromab penetitide for prostate cancer detection, and HIV antigens for diagnosis of HIV infection. Then the nature of therapeutic proteins. So therapeutic antibodies are becoming increasingly prominent in the pharmaceutical industry. Several therapeutic antibodies have been approved for the treatment of cancers, autoimmune, metabolic, and infectious diseases. And monoclonal antibodies are produced by B cells and engineered to target specific proteins that cause disease, are now predominantly used to treat some diseases. Technological advances have also led to the development of biospecific antibodies that can treat complex diseases by um, simultaneously binding to different antigens, and they are being used in the treatment of lymphoblastic leukemia and hemophilia A. So enzymes are an important class of protein therapeutics. Deficient or absent enzymes, as seen in the case of lysosomal storage diseases, can be replaced by enzyme replacement therapy manufactured using DNA recombinant technology. Is currently available for treating 10 different lysosomal storage diseases. Then coming to the types of therapeutic proteins, the first one is antibody-based drugs, which is highly target specific conjugates of antigen-specific monoclonal antibodies and a drug linked through chemicals linkers. And Mylotarg is a commercially available antibody drug conjugated used as an antitumor agent. Then the second one is anticoagulants. Um, which molecules that anticoagulants the molecules that prevent the formation of blood clots. They are administered to patients to reduce the chances of developing strokes and heart attacks. For example, antithrombin, which is a recombinant, is an FDA-approved anticoagulant. Then the third one is engineered protein scaffolds, which is used to develop antibodies with target specificity by providing flexible binding sites for the selection of target molecules. For example, a calantide is an established plasma kallikrein inhibitor. Then the fourth one is interference. Interference, pro and anti-inflammatory cytokines that can actively impact antiviral and anti-tumor effects. Then the next one is interleukins, which is pro-inflammatory cytokines that can be used um, to modulate inflammatory responses in allergy and infectious diseases. Proleukin is a recombinant human interleukin to administered to cancer patients. And enzymes, biocatalysts widely used in industrial and pharmaceutical sectors. Recombinant enzymes can be employed as anticoagulant agents, thrombolytics, antibiotics, anti-inflammatory, and as therapeutics for enzyme deficiency disorders. And also hormones, messenger molecules secreted by multicellular glands. Recombinant hormones are produced and administered to patients with hormonal deficiency disorder, such as diabetes. And many more also are there, like growth factors, um, thrombolytics, F-fusion protein, bone morphogenetic proteins, etc. So here are the applications of therapeutic proteins, including several proteins are created from recombinant DNA, which are also known as recombinant proteins and are used in medical applications like hematopoietic growth factor, interference, hormones, recombinant protein vaccines, tissue or bone growth factors and clotting factors, biological response modifiers, monoclonal, diagnostic, and therapeutic antibodies. So the future directions of therapeutic proteins are that protein therapeutics have an increasingly important role today, and several have been approved for treatment globally. For some diseases, recombinant proteins are the only available treatment, and they can also be used in combination with small molecule drugs to provide additive or synergistic benefits. For example, in the treatment of EGFR positive colon cancer, combination therapy with the small molecule drug irinotecan and the recombinant monoclonal antibody cetuximab results in increased survival in patients with colorectal cancer by synergistically inhibiting the EGFR signaling pathway at different locations. And in the last few decades, efforts have intensified in making the development of therapeutic proteins more economical and easier to produce and in making alternate routes of administration more viable. Given the specificity and range of functions they can perform, therapeutic proteins are poised to play a more significant role in the future of medicine. And the hope is that the protein, which is not present in adequate levels, will function as it is designed to do. For example, use of certain proteins in addressing cardiovascular disease has been evaluated in some studies, especially when veins or arteries become clogged. The right types of proteins might help you by building new passages for blood flow. And some doctors suggest that protein therapy of this type might eventually be so successful that it could eliminate the need for complicated surgeries like bypass surgery.